Hello guys, once again I want to welcome you to our class, and in today's class we want to be looking at reproduction. Reproduction. Okay. Uh I want to start by saying that the need or the desire to reproduce is fundamental to every organism. There is no organism on earth that does not want to reproduce. And the reason an organism wants to reproduce is because it wants that organism wants is life to continue. And when I say life to continue, that means that it gives birth to an offspring that will continue. Sorry, that will continue to carry the same gene or the same makeup in that parent organism. So every organism wants to perpetuate its life cycle or want to continue to exist by extension. And this desire is fundamental to every single organism, including plants, microorganisms, and even animals or macroorganisms. Every organism has this innate desire to reproduce, to birth organisms after their kind, and that is what we mean by reproduction. The process through which parent organisms birth offsprings after their kind. And when we say after that kind, we are a kind of intentional in using that word because you cannot birth organism that is not of your kind. Lizards beget lizards. Lizards beget lizards. Snake begets snakes. And other plants or other organisms will now beget organisms after their own kind. Okay, that is a brief introduction to this topic, reproduction. And then let me just add that uh, it is important you know that organisms utilize lots of resources in order to bring about reproduction. There are, there are lots of resources that are put in place for reproduction to occur. And may I remind you that reproduction takes lots of energy. It takes lots of energy and also takes a toll on the parent organism. And sometimes the parent organism may even lose its life in the process of trying to birth other organisms after its kind. So, in summary, just note as a way of introducing this topic that reproduction is essential to every single organism. There is no single organism that does not have that desire or that does not want to reproduce. The way it is a man, you get to 30, 31, 32 as a man, you want to marry to reproduce. As a lady, 20, 21, 22, you want to marry because you want to reproduce. So that desire is innate. God, imputed this desire in every single organism we have on earth. Having defined this reproduction, then the next thing that I want to look at now is the types of reproduction that we have. We'll have two different types of reproduction, and I will briefly talk about them. The first one is asexual reproduction, and of course, we know we have sexual reproduction. In case of asexual reproduction, you discover that one parent is involved or one organism is involved. It does not involve the union of two different organisms. And because of that, there is no genetic variability. And the implication of not having genetic variability is that a single disease may wipe out the entire generation or the entire population because all the organisms have the same genetic makeup. Unlike in the case of sexual reproduction where there's genetic variability, some organisms may have genes that will enable them to adapt or to survive in such adverse environmental conditions. Then it is also important you know that asexual reproduction has its own pros. I just mentioned one of the pros and one of the cons now. Then other advantages of asexual reproduction is the fact that it is safe. Then it takes less time and less energy. Yes, remember I said when I was introducing the topic that reproduction takes energy. But in the case of asexual reproduction, it's not as complex. The method of reproduction is not as complex compared to that of sexual reproduction. So it takes a lesser time and it also takes lesser energy. So having uh, crisscross Advantages, disadvantages of asexual sexual reproduction. Let's now go into the different types of asexual reproduction and explain them one after the other. The first one is binary fission. The second one is multiple fission. The third one is budding. The fourth one is fragmentation and regeneration. Then fifth one is what's it called again? The fifth one is vegetative propagation. Vegetative propagation. Okay. So in this case now, we're going to be explaining this one after the other. These are methods of asexual reproduction. Methods of asexual reproduction. Now, in the case of binary fission, which is the first one, the word bi means two. So in this case, you discover that the organism divides into two daughter cells. And this division into two daughter cells involves two main stages. The first one is called karyokinesis. Karyokinesis. Then we also have the one we call cytokinesis. Cytokinesis. So this organism has, of course, it's popular in amoeba. Amoeba divide through binary fission. So you discover that this organism, has a nucleus, and this nucleus, when the organism is ready for division, and of course would have matured, because an organism needs to be mature before division begins. So what now happens is that the nucleus divides into two, and once the nucleus divides into two, that is what we call karyokinesis. Karyokinesis, division of the nucleus into two parts. So this nucleus that was previously one will now be two. This is karyokinesis. Then the second kind of division is now what we call cytokinesis. After karyokinesis, a septum is formed there, another septum is formed there, and a wall divides the two organisms into two daughter cells. So this is what happens. This is what happens. A septum is formed and a wall divides the two nuclei to separate them into two daughter cells. So this is what happens in binary fission. Now, the case of multiple fission is similar to what we have in binary fission. The difference is not much. So we also have a cell like this having one nucleus. But then, instead of the nucleus dividing into two, in this case, it divides into multiple parts. You see the nucleus dividing into multiple parts. And if it divides into multiple parts which are separated, then when it comes to cytokinesis, the cytoplasm will also be divided into multiple parts. The cytoplasm will now also be divided into multiple parts. Now divided into multiple parts. So this is what happens in the case of cytokinesis. Multiple fission. So instead of the parent cell dividing into two daughter cells, divides into multiple daughter cells. That is just the little difference between binary fission and multiple fission. It still follows these two stages, karyokinesis and cytokinesis. And like I said, the division of the cytoplasm is cytokinesis. Division of the nucleus is karyokinesis. Then the next stage is what we call the budding. In the case of budding, what happens is that a parent organism, this is the parent organism having its nucleus, develops a bud. It's called bud, bud or outgrowth. If you don't call it bud, you can call it outgrowth. It develops a bud or an outgrowth. Then what you now say is that this nucleus divides into two, and the daughter nucleus migrates into the bud. But before the bud detaches from the parent organism, the bud elongates and becomes mature. mm, just like the human sense, once you become mature as a man, you get to age of 25, 26, 28, you become mature. Then of course you leave your parents' house, you go and look for your own house. That is what happens. So in this case now, once the bud matures, elongates, remember there's already karyokinesis, the same karyokinesis, division of the nucleus, and the daughter nucleus migrates into the bud. From the parent cell, it migrates into the bud, and once the bud is mature, then there's a detachment. There's a detachment of the bud from the parent cell, so that the bud will now begin to live an independent life. So this is exactly what happens in the case of budding. Then we now have the one we call fragmentation and regeneration. In the case of fragmentation and regeneration, an organism, which organism undergoes budding? Of course, Hydra undergoes budding. Then this organism called this protozoa called Toxoplasma gondii undergoes budding, even though it is internal budding, where the bud grows internally. That is called endodiogeny. I don't want to go into that. I think we mentioned that we talked about that in protozoa, in parasitology. Sorry. So, in the case of fragmentation, an organism breaks into fragments. This is popular in fungi. This organism breaks into many fragments, and you can see that this individual fragments uh, can now reproduce of or grow into another mature organism. Each of these fragments, once they fall, on a fertile ground, in the case of a fungi, of course they behave like spores. Of course, you know that once spores fall on a fertile ground, they germinate into that same organism. So that is what happens in fragmentation. So yeast is an example of fungus that does this. Yeast, Saccharomyces, then Saccharomyces cerevisiae, yeast. As a matter of fact, yeast undergoes binary fission, yeast undergoes budding, and yeast also undergoes fragmentation. Then we also have fragmentation and regeneration. This happens in the case of a starfish. If you cut starfish into many pieces, discover that each of these pieces now can regenerate to grow into a full mature or a full-fledged starfish. That is fragmentation and regeneration. Then the last one is the last one. Hope you can see the board. Okay. The last one is vegetative propagation. And this vegetative propagation is peculiar to plants. In this case, you use a vegetative part of the plant to propagate the plant. And the popular one that is always used is the stem. Of course, a plant has three vegetative parts. It has the leaf as a vegetative plant, and of course, the plant can also be propagated using the leaf. A single leaf can birth a full plant. Then the stem, the same thing, the root, the same thing. I wouldn't want to go into that. In tissue culture, that is when this is done. You know, uh, that is where it is done. It is taught in details. So in this case now, the method of asexual reproduction here is vegetative propagation, and mainly the stem is used. So this can also be called a stem cutting. Stem cutting, or probably asexual reproduction through stem cutting. The stem is cut into pieces. Of course, this is popular among people that plant cassava. You see them, they cut the stem or the cassava into pieces. Then each of these pieces is used to regrow a full mature cassava. That is vegetative propagation. So it's peculiar to plants. So these are methods of asexual reproduction that we have. Methods of asexual reproduction that we have. So having crisscross methods of asexual reproduction, the next thing we're going to talk about now is mitosis and meiosis. Mitosis and meiosis. if you can agree with me that mitosis and meiosis are two different topics, mm, that students find difficult, and it's difficult to see a teacher that will, explain these two processes in such a way that students will understand. That is why students run away from it. They feel it's too difficult. So we're going to x-ray that, the mitosis and meiosis, in no distant time. But then let's begin with what we call the cell cycle. Let's begin with what is called the cell cycle. Okay, the cell cycle. Please note that cells divide on regular basis to bring about growth and reproduction. So for cells to grow, uh, they undergo this cell cycle or this division. They divide on regular basis to bring about growth and reproduction. Not just growth, but even in reproduction. That is one of the essence, or one of the reasons why cells divide. Why cells divide? In terms of growth, the more cells divide, the more the organism grows. Let's assume that that is talking about mitosis now. You have, of course, the mitosis occurs in the somatic cells, whereas meiosis occurs in the reproductive cells. That helps in birthing of the sperm and the egg. That is for meiosis. Then let's assume that you had a wound on your skin. This is a somatic cell. You have somatic cells on your skin, and a space has been created. So to replace the damaged cell, mitosis has to occur. Then the cells will now begin to divide. The more they divide, the more they cover the space. The more they cover the space, and that's one of the processes involved in healing of wound. The cells have to regrow back. And of course, mitosis also occurs even in after reproduction. Once a zygote is formed, then mitosis also has to occur. That is when you're now studying organogenesis and spermatogenesis and the rest of them, which we are still going to do if we have time. So, but the essence of this topic now is to describe the steps involved in mitosis and the steps involved in meiosis. So, in the case of cell cycle, just like I said, that organisms divide on regular basis. They divide on regular basis in order to uh, uh, grow and to also reproduce. This division occur in a sequence of events that is now called a cell cycle. The divisions I just mentioned occur in a sequence of events. This sequence of events is called the cell cycle. The cell cycle. And I want to describe the cell cycle. The cell cycle. The cell cycle is like this. This is a cycle. Then we'll have this, we'll have this, we'll have this, we'll have this, we'll have this. This is called the G1 phase. G means gap. So this is gap 1. Then we'll have S, we'll have G2, then we'll have mitosis. Mitosis. From this line down to this place is what we call the interphase. The interphase. So people have been making mistake in the past, and that mistake is that they think interphase is one of the stages of mitosis. That is very wrong. And I've seen that in some textbooks. That is very wrong. Interphase is different from mitosis. As a matter of fact, the cell cycle has two different phases or two different stages. We'll have the interphase, we'll have the mitosis. Until these two are completed, a cell will not divide. So the mitosis begins with G1, S, and G2. So in G1, that is when the cell, begins to prepare for division. When a cell begins to prepare for division, that is what happens at G1. And how does a cell prepare for division? That is when the cell begins to synthesize other cellular components. Remember that this cell wants to divide. And just like I mentioned in binary fission, it divides its nucleus, give part of it to the daughter cell. In this case, the different components that make up that cells have to be divided. Eh? Have to be divided because another cell will be bettered from that single cell. And that is why there is need for cellular components to to be synthesized. And not just that the cellular components are synthesized, the cell also begins to grow, because we are still going to talk about limitation or regulation of cell division, and cell size is one of the factors that regulates cell division. So after this stage now, we now have the S stage. The S stage is the stage of DNA replication. This is the word S means synthesis. Synthesis. So this is the stage of DNA replication. So DNA replication occurs here. DNA replication. DNA replication. Then at this G2, the cell continues to grow. The cell continues to grow. This, I've said what happens in G1, then what happens in S, then in G2, the cell normally continues to grow. In G2, continues to grow. And of course, okay, let me not say that. I think that happens in prophase. So the cells continue to grow, to grow again. It has grown in G1. It started growing in G1, then DNA replication, then it continues to grow because, like I said, cell size is one of the factors that decide if a cell will divide or not. So a cell should have grown to a certain level before division occurs. Then it is also important I point here that it is at the G1 phase that a cell undergoes a check. There's something like a checkpoint at the G1 phase. It is within this checkpoint or at this G1 phase that is when the decision to either divide or not to divide occurs. The decision to either divide or not to divide occurs at G1 phase. So a cell, if all the necessary cues, nutrients, and resources are available, and of course the DNA is okay, I'll still mention some of these things again, then that is when the cell now enters into the S phase from the G1 phase. But at this G1 phase, if all the necessary factors necessary required for cell division is not there, then of course the cell will now return back to the resting phase or probably take the decision not to grow. There is a phase which I didn't write here that is called the G0 phase, the gap zero. Now this is what we call the quiescent phase. If a cell, sometimes cells can enter into this quiescent phase temporarily, like the liver cells, sometimes they enter into this quiescent phase and don't divide again until when there is need, probably there's an injury, then uh, a kind of changes from this G0 to G1. But there are many cells that enter this the G0 phase, the quiescent phase, and they don't divide again. Something like the muscle cells, something like the nerve cells, they enter this phase, and this is a phase of no division. Permanently does not divide again because the nerve cells do not have any need to continue division. Once they are formed, they are formed. The muscle cells do not have any need for division. Once they are formed, they are formed. So these are the ones that enter into the G0 phase permanently. But the liver cell can enter this G0 phase temporarily, after which, if there's need, then it gets into this G1 phase and the cycle continues. But like I said, there are so many factors that determine if a cell should divide or not divide. And that's proofreading of the preparedness of a cell to divide or not to divide is done at the G1 phase. If the necessary materials, the factors that are necessary are there, then of course it kicks off from G1 to S, then to G2. But if they're not there, then the cell turns off and will not divide until when those resources are found or when those factors are available, then it can now enter and begin division. So this is what happens in the what? In the this is what happens this is what happens in the cell cycle. Then we'll now go into the main one, the main one, which is mitosis. We have talked about these processes, and all this has happened within the interphase. And I forgot to mention that energy is also generated within this interphase. There's energy that is generated, ATP, because division. You see, reproduction takes lots of energy, like I said. It takes lots of energy. As a matter of fact, all the metabolic processes within the cells are powered by energy in form of ATP, the energy molecule of the cell. If this energy is not found there, if this energy is not there, then of course the cell cannot divide because everything is energy. Everything is being powered by energy. Every metabolic process within the cell. So especially this one. So one of the things that also happens at interphase is that as a cell is preparing, generating or reproducing or synthesizing other cellular components that are important for this process, then energy is also produced at this moment in form of ATP. All right, so let's now go into I hope this is okay. Let's now go into uh, mitosis. Mitosis. I've cleared the misconception that interphase is not a stage of mitosis. I've said everything that happens at the interphase stage. So I won't mention interphase again. Let's now go into the different stages that we have. We'll have the prophase. We'll have the prophase. We'll have the metaphase. We'll have the metaphase. We'll have the anaphase. We'll have the telophase. Telophase. So these are the different stages of mitosis. The different stages we have in mitosis are these: the prophase, the metaphase, the anaphase, and the telophase. Okay, so since you have known this, so let's clean the board because this is where we're going to. So let's look at this. So this is a cell. This is a diploid cell with homologous chromosomes. Members of a pair of a chromosome. This is a pair. What's happening? Members of a pair of a chromosome is said to be what? Homologous. Members of a pair of a chromosome. Sorry, members of a pair. Yes, members of a pair is said to be what? Homologous. So these two make up a pair. These two make up a pair, and they're said to be homologous chromosome. Then, of course, this is a sister chromatid. 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 Okay. Now, this cell now wants to go into the division, and this is the original chromosome or number of chromosomes you see here. Let me draw this in turquoise again. So this homologous chromosome, and of course, they are held together by centromere. They are held together by centromere. So you have centromere here, or have centromere here, holding each pair of this chromosome. And like I said, one from the mother and one from the father. This is sister chromatid, is sister chromatid. These two are non-sister chromatid. Then these two are homologous chromosome because they are members of a pair of a chromosome. They are said to be homologous. Then after DNA replication, and like I said, this DNA replication occurs But there's a reason I'm bringing it up here again. But please note that DNA replication already occurred at the interphase stage. It is not occurring here, but there's a reason I'm bringing the original cell before division and after DNA replication for the sake of emphasis. So after DNA replication, this one replicates into two, this one replicates into two, this one too replicates into two. So this is what happens. Then this is now the the cell or the number of chromosomes you're going to see after replication. Then after replication, okay, the after the prophase, of course, what happens at the prophase? In the prophase, that is when the spindle fibers are formed, and of course, you know the function of the spindle fiber is that it helps to hold the chromosome. It helps to hold the chromosomes, and these spindle fibers, sorry, these spindle fibers are produced by the centroids, but plants do not have centroids. So how do they produce their own spindle fibers? They have what they call the microtubule organizing center. Microtubule, microtubule organizing center. The microtubule organizing center, this is what the plants use in the production of their spindle fiber because they don't have centroids. Okay, so during the prophase, the spindle fibers are formed. Then you now discover that the nuclear membrane breaks down. The nucleus also dissembles. That means also breaks down. And some textbooks will say disappear. It's not like the nucleus disappears because where is it disappearing to? But the components that make up the nucleus, like the genetic material, the proteins, they break down in such a way that they become invisible. They become invisible. They become invisible. And another thing that happens in prophase is that that is important. I almost forgot. The chromosome, when the chromosome is at the stage of interphase, non-dividing, is usually in the form of a thread of DNA and protein. And this kind of protein, of course, is called histone in eukaryotes. The kind of protein that attaches or that binds the DNA is called histone. So when the cell is in non-dividing state, the chromosome is in an invisible or in an uncondensed form or an invisible form that is called chromatin. That word chromatin, that it means a chromosome that is uncondensed, that is not visible, is just mixed up in such a way that they are not clearly divided. They are not clearly divided. But in the case of prophase, that is when the chromosomes now condense. There's condensation of the chromosome, then there's breakdown of the nuclear membrane, then there's also breakdown of the nucleus. And like I said, the spindle fibers are formed. So these spindle fibers are formed. This is a diploid cell, that is undergoing division. Then after replication, this is what that happens. And I've explained what happens in prophase. So let's now go into the metaphase. Now, at the metaphase, what now happens at metaphase is that the chromosomes now begin to align themselves at a place we call the metaphase plate, or you call it the equator of the spindle. The equator of the spindle or the metaphase plate. So you see the chromosomes aligning themselves here, aligning themselves here. Then this is what happens at metaphase. Metaphase. Then we'll now get into the next stage, which is anaphase. And the anaphase, what happens is that the chromosomes are now being drawn at the opposite pole. They are now being drawn at the opposite pole. The chromosomes, you can see that the chromosomes are now being drawn at the opposite pole. So this is what happens at anaphase. At anaphase, they are pulled apart. They are pulled apart with the help of the spindle fibers. They are pulled apart into the opposite poles. And of course, at telophase, that is when cytokinesis occurs. And cytokinesis is a division of the nucleus into two daughter cells. Cytokinesis. And once cytokinesis occurs, the spindle fiber, because we don't need the spindle fiber again. It has performed this job. The spindle fiber breaks down. Then that nucleolus that once disappeared, quote and unquote, you remember what I mean now, that once disappeared, reappears again. Then the nuclear membrane that breaks down reforms again. And that is the end of mitosis. You can see that we have two daughter cells. And these two daughter cells have the same number of chromosomes with the diploid cell or the parent cell from whence this division began. It has the same number of chromosomes, the same diploid number of chromosomes, 2n number of chromosomes. So this is how it happens in the case of mitosis. So let's now go into the second phase, which is meiosis. Meiosis is more complex. Meiosis is more complex compared to meiosis is more complex. So in the case of meiosis, we have two different phases. Okay. So in the case of meiosis, we have two different phases. We have meiosis 1 and meiosis 2. We have meiosis 1 and meiosis 2. Just a minute, let me get marker. So we have meiosis 1 and 2. We have meiosis 1 and meiosis 2. So we're going to look at them one after the other. Let's start with meiosis 1. And let's look at what happens in meiosis 1. In the case of meiosis 1, the same thing that happens, I also want to mention interphase here. The same thing I described in the case of interphase is also similar with what to happen. The same thing I described in the interphase of mitosis is also the same with the interphase of meiosis. There's no difference. That is for interphase. But then, okay, sorry. Yes, that's the same thing now. The same thing that I described in the case of meiosis, mitosis is the same thing with meiosis. The interphase is the same thing. That is when the cell prepares to divide and all that. The G1 phase, the S phase, and the G2 phase. So we will now go into the prophase. And please understand that the prophase is the longest phase when it comes to cell division. The longest phase in cell division is the prophase. And let's now look at what happens in prophase. We'll have prophase 1, we'll have prophase 2. So under this meiosis, we'll have prophase 1. And there are different things that happens in prophase 1. Prophase 1 has many stages of prophase 1. Prophase 1. We'll have the first one, which is called the leptotene. We'll have the Zygotene. We'll have the Pachytene. We'll have the Diplotene. We'll have the Diakinesis. So let me quickly describe what happens in all these stages. So in the first stage, which is leptotene, that is when the chromosome begins to condense. Remember, at interphase, the chromosome is at a state that is uncondensed, is invisible. Then at leptotene, the chromosome is still thin. It's still thin because it's just coming out from the interphase. is still thin, but begins to condense a little. And that each of the chromosome in this case has two sister chromatids. After replication, that is what it occurs. That is what happens. By the time I get into the rate process, we'll look at that again. So each of the chromosome will now begin to have sorry, a minute. So like I said at leptotene, there is condensation that begins to occur in the chromosome. They form thin thin thread. The chromosomes form thin threads and they begin to condense little by little. And each of the chromosomes now have sister chromatids because they have replicated. And that replication occurred at interphase. Then zygotene. What happens in zygotene is zygote, just like a zygote, pairing. So there's pairing of the homologous chromosomes. The homologous chromosomes will now pair together. And the pairing of homologous chromosome is what we call synapses. 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 As a matter of fact, they form what is called they form a complex called synaptonymal complex. The homologous chromosomes form what is called synaptonymal complex. Apart from the chiasmata that holds the homologous chromosome, there is another region. There's another region within the body of so this chiasmata holding this homologous chromosome. Apart from this chiasmata, within the body of this chromosome is within the body of this chromosome, these homologous chromosomes on the way it is drawn, you know, I mean on the board is not exactly the way it is. And there's no how I can draw that synaptonymal complex. It's difficult to draw, except you Google it, you see the picture. There are a kind of fine traits. There are kind of fine traits that hold, if this is a homologous chromosome, this is another homologous chromosome. There are fine thread-like structures that hold them together. Apart from the junction where they are being held by the chiasmata, apart from that junction where they are being held by the chiasmata, there is another thread-like structure that holds them on their body. And that is what we call the synaptonymal complex. Because it's a complex, it's made up of so many threads holding the two homologous chromosomes together. So the pairing of homologous chromosome is what we call, it happens at the stage called zygotene. And that process is called synapsis and they form synaptonymal complex. The third stage now is Pachytene. In the case of Pachytene, look at what happens. What happens that is when that crossing over. I know you might have heard about crossing over in meiosis. Crossing over in meiosis occurs in prophase 1 of meiosis. And the actual stage in prophase 1 is in the Pachytene. That is where the crossing over occurs. In this crossing over, that is when the homologous chromosomes now exchange their segments. The exchange of segments between homologous chromosomes now occur within this Pachytene. Then in Diplotene, something happens. And that thing that happens is that this complex begins to break down. The synaptonymal complex begins to break down, but then the homologous chromosomes are still being held together at the chiasmata. But the bond holding them between this or in the synaptonymal complex begins to shift or begins to break. However, the bond holding them at chiasmata is still there, still very strong. The one that begins to shift or break is this synaptonymal complex. That is what happens at Diplotene. Then diakinesis. At diakinesis, that's the last stage of prophase 1. That is when the cell is now ready to begin the process of meiosis, and then that is when the nuclear membrane breaks down, the nucleolus disappears or breaks down, then the spindle fibers will now begin to be formed through the centroids. Okay, so that is what happens. So invariably, I've explained prophase 1 of meiosis. So let me now start with So let me now start with the metaphase. Since I've explained, remember that this is after replication. Before replication, this is a diploid cell. Before replication, this is the diploid cell having this homologous chromosome. Then after the replication or duplication, this one produces these two, this one produces these two. So this is what we'll have. And I've explained all the stages that happen, all the stages. So what you'll now see is that, okay, let me draw this. Because because crossing over has occurred, or because synapses has occurred, you are going to see that these chromosomes are now held to each other. Synapses has already occurred in prophase 1. So the homologous chromosomes are still being held by each other. Remember I said that the synaptonymal complex begins to break, but then the bond holding them at the transmatter is still there between the homologous chromosomes. So what now happens is that this is what happens. Then let me now describe what happens at the metaphase. At the metaphase, remember that at the metaphase, that the homologous chromosomes now align themselves at the equator of the spindle fiber. They align themselves at the equator of the spindle fiber. At the equator of the spindle fiber. And remember, they are still being held together. Don't forget that they are still being held together. So this is what happens in metaphase. Then after metaphase, we now have anaphase. So in the case of anaphase, you now discover that they are being drawn apart to the opposite poles. They are being drawn apart to the opposite pole. Look at this carefully. This is when the bond between the homologous chromosomes are completely broken. The synaptonymal bonding, just a minute. So this is where the bonds between the homologous chromosomes are completely broken. The synaptonymal complex, remember I said, begins to break at where did I mention that? I mentioned that at Diplotene. But now at this stage, that is when they break completely. And apart from the fact that they break completely, the transmatter, the bond holding them at transmatter is also completely broken. And don't forget that crossing over has already occurred. So there's no need of the chromosomes or the homologous chromosomes being still held together, because they have achieved the aim of coming together. And that aim of coming together is for what? Crossing over. So crossing over has already occurred. So in this case now, since the bond is broken, the chromosomes now go their separate ways to the pole, to the opposite poles. So you see this chromosome here. So you see this chromosome here. This one goes to this opposite pole. This one goes to this opposite pole. Remember that these two were attached together by the synaptonymal complex and by the transmatter, but they are now being separated to the opposite poles. The same thing happens to this one. They are also being separated and they go to the opposite poles. They're also being separated and they move to the opposite pole. This is what happens at anaphase. Then the last stage is telophase. Then at telophase, That is when at telophase, that is when uh, cytokinesis occur. Remember, these are already at the opposite pole. This is already at the opposite pole. Then cytokinesis occur. And when I say cytokinesis, there's a breaking of the cytoplasm into two daughter cells. Breaking of the cytoplasm into two daughter cells. And there's something that happens here. There's something that happens here. The spindle fiber has performed this function. So at this level, at this stage, it breaks down. The nuclear membrane that broke down before reappears. The nucleolus that broke down before also reappears. And this signals the end of of meiosis 1. This signals the end of meiosis 1. We are going to now go into the second stage of meiosis, which is called meiosis 2. Just a minute. All right. So let's now go into, this is what we have. This is a product. This is a product of meiosis 1. This is a product of meiosis 1. So we're going to start with this product of meiosis 1. We're going to start with this product of meiosis 1. Let's draw this product of meiosis 1. So this meiosis 1 has given us two daughter cells. Two daughter cells. So now I have two daughter cells now. This is two daughter cells. Don't forget that mitosis gives or produce offspring that have exact number of chromosomes with the parent. But there's halving, what we call halving of the chromosome to give haploid chromosome or to form haploid chromosome in the case of meiosis. And that is what we call reductive division. There is a kind of reduction of the chromosome, the number of chromosomes seen in the parent organism, and we are going to see it now. So these two cells are cells that are formed at the prophase 1. I hope this thing is not too long. All right. Let me quickly say something before I continue. There's something we call the interkinesis. Interkinesis. Now, interkinesis is a short rest that is that these two cells, that is product of meiosis 1, enters into before they now begin the second stage, or before they start the second stage. Just like you are running to a place, you have not gotten there, you stopped at a place and you're just resting. Trying to rest before you now start the second stage. But don't think that DNA replication occurs again. No. The DNA replication has already occurred in the interphase. It's not occurring again here. So the division continues with the cells that are already produced. Okay, so look at what happens in this case now. So this is I've explained the interkinesis. That is where after telophase 1, the cells enter into a short period of rest before they now begin the journey again. And this journey will now begin with prophase. But all these other things I mentioned in prophase 1, we are not mentioning it again. All the things I mentioned in prophase 1, we are not mentioning it again. So what happens now in this case that in prophase 2 is that the spindle fiber that broke down in telophase 1 will reappear again or will reform. The centroid begins to produce another stage of spindle fiber. We are not going to use the same spindle fiber we use in meiosis 1 for meiosis 2. So a new set of spindle fibers ought to be produced. And once they are produced, now they are produced at prophase 2 of meiosis. And the other thing that happens is that the same nuclear membrane that formed in telophase 1 will break down. The nucleolus that also formed will also break down. These are the things that happen in prophase 2. So let's now get into the second stage, which is meiosis. Sorry, metaphase, which is metaphase. Metaphase. So the case of metaphase, just like we have explained before. So you see that these homologous chromosomes, they they align themselves at the center of the equator. They align themselves at the center of the equator, aligning themselves at the center of the equator. Hmm? They align themselves at the center of the equator. Then this is for metaphase. Metaphase now. This is for metaphase. Then what now happens? What now happens is that this is metaphase. At anaphase, the chromosomes are drawn at opposite pole. At opposite pole. So the chromosomes are now drawn at opposite pole. So you see that this one goes to the opposite pole. So the same thing happens here. Opposite pole. Then of course, telophase 2 and telophase 2, telophase 2. So at telophase 2, you now discover that cytokinesis occurs. And once cytokinesis occurs, the spindle fibers are broken down, the nuclear membrane reform, the centroids, the nucleolus also reform. The nucleolus also reform. Then there's something I've not mentioned before. Once telophase is done, these chromosomes that were once visible will now decondense. In case I didn't mention it in mitosis, remember, once we are done, or once it gets to telophase, the chromosomes that were once visible will now become invisible. They will now do what we call decondensing. They will now decondense. And as they decondense, they enter the resting stage, which is interphase. And when next the cell has all the necessary resources and the enabling factors to divide, the cycle continues. So this is what happens in meiosis. You can see why I retained this parent organism before we started this journey. I retained this parent organism to show you that this cell now, these cells now have half. These cells now have half. Half of 2 is 1. Can you see 1 is here? Instead of having 2 cells per cell, we now have 1. So at the end of meiosis, we have 4 cells. 1, 2, 3, 4. And each of them having half of the diploid number of chromosomes found in the parent cell, from whence the division started. But in the case of mitosis, you discover that the number of chromosomes were exactly the same with that of the parent organism. So this is what happens in mitosis and meiosis. And I hope I made it clear, because I know lots of students and even teachers are running away from this topic, mitosis and meiosis. So before we go finally, let's now talk about the regulation in cell division. The regulation in cell division. Cell division has lots of so we have factors that regulate or determine how cell it either enhances the process or inhibits the process. Researchers have studied cells in culture, cultured some cells and now found out that there are some toxins or some poisons that can inhibit cell division. Yes, once the toxins are available or are within the confines of within the environment of that cell, then it inhibits cell division. Apart from poison, remember I said factors that regulate. So these factors can either help to facilitate the process of cell division or inhibit the cell division. We have growth factors. Growth factors also contribute to cell division. If growth factors are not available, then of course cells will not divide. Like the fibroblasts, the cells that are found in connective tissue that help in the in healing of wound. This cell has what they call the platelet-derived growth factors. They have platelet-derived growth factors. So once these growth factors, the platelet-derived growth factors, once they are available, then the fibroblasts will now divide quickly. And of course, this helps to facilitate healing of wound. Then it is also important you note that cell size is also crucial when it comes to cell division. A cell cannot divide if it does not grow to a certain size, and that is what happens at the interphase stage. A cell must have grown to a certain size before the cell begins to divide. Because if the cell does not grow to a certain size, then the multiple division will make the cell to be too minute. We made a cell to be too minute, and that is what happens in the case of cancer cells. Cancer cells do not undergo some of these things. I'm trying to. Cancer cells, they run their own parallel government. Sorry to use that word. They run their own governments. They run parallel government differently. So cancer cells don't obey some of this regulation, don't come under this regulation. So cancer cells do not even grow to certain size before they begin to divide. And that is why you see that they are so minute. Cancer cells are so minute compared to normal cells. If a cell does not grow to certain size before it begins to divide, then a time will come when that cell will be so minute. It will be so minute, so minute. Imagine a cell divides, then that one divides. A time will come when the cell will be gone. And that is why a cell needs to divide and increase its size. And what you use to measure the size of a cell is the ratio between the cytoplasm and the gene and the genome. The cytoplasm and the genome. Of course, the size of the genome remains the same because it replicates. But that of the cytoplasm, there needs to be synthesis of substances to beef up the one found in the parent cell before the cell begins to divide. So the ratio of the cytoplasm to the genome is what determines what is used to determine if a cell has grown to a normal size before division. Then another factor that can also inhibit cell division is what we call the contact inhibition or the density dependent inhibition or density dependent factor. The density of a cell, the number of cells that are found within uh, the cell, or let me say crowding, crowding also inhibits cell division. And this is one of the mechanism that is found in normal cells to regulate the way normal cells divide. And this mechanism too, is also lost in cancer cells because cancer cells do not have this mechanism. Crowding does not limit the growth or the division of cancer cells, but it limits the growth or division of normal cells. So when cells become crowded, and this was studied in an artificial environment using petri dish. So if we have this as a petri dish, and these are the spaces within this petri dish, these cells are growing and they are growing and they are growing, multiplying. Once they cover the whole surface, they stop. Except probably a space is created here and they will now grow again to fill up that space. And that is what happens in the case of human. Assuming you have a wound during mitosis, then what the cells do is that they multiply and grow to cover up that wound. And once they cover it up, they stop dividing. So cells divide when they have the necessary signal and a normal environment. So if the cells are divided to a level that they have covered the environment they are supposed to cover, because they form a thin layer. Cells normally, when grown in artificial medium, they form thin layer of cells, or single layer of cells. Cells don't grow on top of each other. They just grow on the surface of the container. Once they occupy the surface of the container, then the cell starts growing. So crowding is one of the check system that regulates cell division, but it does not happen in cancer cells. Then, like I said, this density dependent factor is also called contact inhibition. Then, of course, I mentioned growth factors that also determine cell growth. Then, DNA integrity is also an important factor. If there is a break, a break in the DNA, probably during replication, or maybe at any time, then the cell will not divide. So that break or breakage of DNA can either make the cell to pause until something is done about it, or possibly to stop dividing, completely. But again, cancer cells do not obey this. They continue to divide regardless the integrity of the DNA. Then nutrient availability. So if nutrients are not available, cells will also not divide, because cells require this nutrient to divide. And like I said, this is being checked at the G1 phase of the interphase. It is being checked to see if all the necessary cues, all the necessary resources are available for the cell. If they are available, then the cell continue to divide. But if they are not available, then the cell will stop dividing. Ladies and gentlemen, I think with this viewpoint of mine, I think I've done justice to this reproduction with emphasis on mitosis and meiosis. See you in my next class. Please, if you have not subscribed, try as much as you can to subscribe and share this video. I know you may have people all over the world or different universities in Nigeria that may need this teaching. So be kind enough to share to them. I wish you the best of luck. See you in my next class.